Started again. I'm Stefan Dever. I'm one of Emily's team board of advisors. So this is Emily Nordin is going to talk about the work that she's done. Emily is a crazy person. She studied 4 majors here at Lawrence: chemistry, biochemistry, English, and is French. I always want to say history. Right, French. And she can, because she is the bravest student that I've seen in some time. So oftentimes I talk with sophomores about research opportunities both on and off campus. I had a conversation with Emily like that toward the end of her sophomore year. And at the end of these conversations, I usually say something like, you know, if you can't get one of these prestigious REU positions, you can just cold call somebody and just try to talk your way into a job. Because I've seen that happen. Not very often, but it's possible. And then maybe a week later, Emily said, I found a person. I'm like, great. I imagine she would just go down the street, somewhere in the state. And she said, I'm going to Scotland. And I said, why? And she said, Scotland is cool. And so, okay, and my God, she did too. She made it work and got hooked up with an amazing research group in Scotland and did amazing work and explained it well and came back and did a little bit of the work.
Some hand waving there.
Well, there's some mind to it. She took it back to my lab, taught me about the chemistry that she was doing. We worked out some of the how to think about it. She went back for the next summer and did even more amazing work. And it was really neat to see her famous independence applied for this really amazing opportunity. So without further ado, I want to hear more about Scotland. So let's welcome her.
So hello, my name is Emily Nordin, in case you don't know me. Today I will be talking to you about the research I did at the University of St. Andrews in Scotland with the Westwood Group in the summers of 2014 and 2015. The title of my talk is Towards the Target Identification of a Bioactive Compound in Cryptosporidium parvum. There are a couple things you might notice about this title. First off, there's an S in the word towards. And I've been told by many people that that's not how you spell the word toward. In fact, that is how you spell the word towards in the UK. So keeping it. Just the way we're going to do it. The second thing you might notice is that this is a very jargon-heavy title. So we're just going to skip over it for now. And move on to an easier question. So the question I want to pose to you guys is, how do you develop a drug? Maybe some of you have some idea of how it works, and if not, it's okay, because we're going to talk about it today. So this is an easy question to ask, but the process behind drug development is quite complex. It's rather discouraging, very time-intensive, and above all, it's super expensive. So this is the drug development process. As you might notice, only 1 out of every 10,000 conceptualized drugs actually make it to the market, which is rather abominable. You might also notice that it takes about 12 years for this process to happen, like Charlie had mentioned before. And the latest numbers show that it takes about $2.6 billion to bring that drug all the way to the market. Which, to put this in a little bit of context for you, President Obama has recently asked Congress to allocate about $2 billion to fight the Zika virus outbreak that's been happening. The House has only allocated about $500 million. So knowing that it takes $2.6 billion to get a drug to the market, do you think that it's likely that we'll be able to develop a drug for Zika soon? Not to be alarmist, but probably not. We're going to start with the first stage of drug development, is known as drug research. Or to those in the know, it's drug discovery. So in drug discovery, essentially you take a bunch of compounds and you test them against your pathogen of interest. And you see what happens. And see if there's any activity against that pathogen. If the pathogen dies, or if the pathogen lives and prospers, whatever it happens, you want to know what's happening with that interaction there. The next stage involves preclinical trials, whereby you take your potential drug and you feed it to a bunch of monkeys or some animal, and you see what happens. You see if they die, and if they don't, you can move on to the next stage, clinical trials, where you do the same thing again with humans, see if anyone kicks off or dies. Hopefully that doesn't happen. And if it all works out, you move onto the fourth stage, where you get approval for your drug, and then you can bring it to the market, and from there people can buy it. And then this last stage, which is kind of optional, depending on the drug that you're developing, you can continue to research that drug and how it works in the body after it's been to the market. So for my talk today, I'm just going to focus on this first stage of drug development, known as drug research or drug discovery. There are 3 steps involved. We're going to walk you through, there's no here. I'm going to walk you through each one of them. So here we go. So first, you choose a disease of interest. Second, you choose a lead compound, just another name for a potential drug. And finally, you identify the biological targets, in our case, the protein targets of that lead compound of that potential drug. So here we go. The first step. Choose your disease of interest. How do you decide what this is? It's kind of a hard question to some people, and for some people it's not. So you might be motivated for personal reasons. Maybe someone in your family has a disease and you've decided to make a drug for it. On the other hand, you might be motivated for financial reasons, because you know a lot of people are sick in the world, and as history has shown time and again, it's very easy to profit off of human misery. Some combination of these two, financial and personal benefit, or personal motivation, you're going to decide on what type of disease you want to study. So the disease that my group studied is cryptosporidiosis. It's caused by the parasite Cryptosporidium barbum, also known as C. barbum, also known as crypto. It's a waterborne pathogen, and I'll walk you through the life cycle right here. So first, you ingest contaminated water with that parasite, or somehow you're swimming in it, like in a pool of some sort, and that gets into your body. You ingest it, and then that parasite will go down into your small intestine, where it will wreak havoc. Then that parasite will be excreted from your body, and the cycle will continue. So cryptosporidiosis affects about 7,000 people per year in the U.S. That might seem like a really small number in comparison to other diseases in the world. But actually, in 1993, there was an outbreak in Milwaukee where 400,000 people were infected with this parasite within the span of 2 weeks. The Milwaukee public school system had to dump out hundreds of pounds of Jell-O because they made it with contaminated water. And if that weren't tragic enough, 69 people died, and 93% of them had AIDS. This is just a picture of some woman who is boiling her water to get the parasite, kill the parasite. So symptoms of cryptosporidiosis include watery diarrhea, nausea, and fever. This is definitely a problem for people who are immunocompromised, like people with AIDS. It just exacerbates their problems. Current treatments are limited in efficacy. They only work for some people, and when they do work, they don't work that well. And no preventative vaccines actually exist, so that's what kind of the motivation for my group sitting down and studying this. So, to recap, we've just chosen our disease of interest. So our second step is to choose a lead compound, our potential drug. So lead compound, potential drug, a chemical compound that shows some bioactivity against a pathogen. Again, that activity can include inhibiting that pathogen in some way or supporting, promoting that pathogen. Bioactivity can be quantized. One variable or one measurement that we use is called the half maximal inhibitory concentration, also known as the IC50. and that's the concentration of that lead compound that it takes for to inhibit 50% of the pathogenic activity. So here's a nice little graph. You can see as concentration increases, you have more increased inhibition, and where you have 50% inhibition, you have your IC50 value, corresponds to your IC50 value. So how do we even determine what our lead compound is? One method to do this is called high throughput screening. Essentially in high throughput screening, it's an automated process. You just screen or test a bunch of potential lead compounds, potential drugs against your pathogen of interest. As we can see in this picture here, it's automated but with a machine. Essentially, you take all of your lead compounds, potential lead compounds, you throw them into a well plate, you throw in your pathogen, and you see what happens. But it's a bit of a sticking point with high throughput screening. It's called high throughput. You need a lot of compounds to test. And the sad thing about chemistry is compounds don't grow on trees. They're very hard to come by and they're very difficult to make. Except, of course, if you're using compounds in a leaf, I guess. So my lab group had to find a database of some sort filled with hundreds of compounds to test. So we used the malaria box, like Charlie talked about. Malaria box is a database of 400 potential drugs, potential lead compounds that exhibit activity against Plasmodium falciparum, which is one of the main malarial parasites. Why are we talking about malaria if I just was talking about cryptosporidiosis? It's because this is just a database and you can test these compounds against all sorts of different parasites. It's just a good way to get a lot of potential drugs quickly. So here it is. It looks like it's in a nice treasure chest here. In fact, it's just like this clear, a lot of little clear boxes, which is pretty sad. Actually, I just looked it up. The malaria box is open source, so anyone can get it, as long as you publish your results and let other people see your results. Unfortunately, malaria box is no longer open. No one can get it anymore for reasons unknown. Probably it has something to do with money, because it takes a lot of effort to synthesize 400 compounds. So we had to choose our lead compound, so we tested, or my lab group did, tested all the 400 compounds in the malaria box, and they chose one that had good activity, a good IC50 value against Cryptosporidium arba. So this molecule is known as a quinacylate. Some points to note is that it has 3 ring systems. You have one system here, another one here, and another one on the bottom. And you have 2 NH groups, which are always fun to have in a compound. So, let's recap. We've chosen our disease of interest, cryptosporidiosis. We've chosen our lead compound, known as MMV006169. And now we have to identify the biological targets of that lead compound. So essentially, what we are looking for, we're just looking to see how our lead compound is interacting with our pathogen and how that pathogen is going to crump and die. So it might be a good time to ask, why do we even need biological targets to begin with? Well, it all comes back to specificity. So, if you might know, back in the 1960s and 1970s, chemo drugs were kind of coming onto the market. And the problem with those chemo drugs is that they weren't very specific. They had a lot of different biological targets in the body. So when you were undergoing chemo, the drug would kill both your good cells, the immune cells, and would also kill your bad cells, your cancer cells. So it basically just savaged your body, which is bad, very toxic. So we want to find a way to identify the protein targets, the very specific protein target of our lead compound in C. parva. There's a lot of different methods to do this. The one we're going to focus on is the yeast-free hybrid method. So there are 3 components, as you might suspect, with the yeast-free hybrid method. First, the first component is comprised of a hook, which involves using a reporter gene, which is going to tell you if that interaction between your lead compound and your pathogen actually happened. You We'll also have a DNA binding domain, which recognizes a specific DNA sequence on your reporter gene. And finally, you will have some random known protein, which isn't important for us. The second component is known as the bait component. It has a known ligand that will bind with that known protein. It's not very important, so put it out of your mind. The second component will be a linker. That's a huge molecule. It's like 100, some atoms. It can be. It's very big. And then you will have a lead compound. Again, this is for us. It's MMB006169. And finally, you will have your fish, which is comprised of that unknown target protein that we really want to study in C. parvum, and an activating domain which allows proteins to bind to it. This whole thing acts like a transcription factor, activating transcription, turning on the gene. And this all takes place inside a yeast cell. So here we go. We can look at this a bit more closely. We have our reporter gene. We have our DNA binding domain, which is going to bind to this gene at some point. And we have a just a random protein. Then we have our bait. Again, we have a ligand that binds with this protein. We have a linker that connects the ligand with the lead compound. And then finally, we have our fish component, which is comprised of the protein target and the activation domain. So when the hook, bait, and fish components all come together, the reporter gene is expressed in the cell. Cells that express the reporter gene undergo a physical change of some sort that researchers can detect. And from that point on, we can take that cell out. We can take out the DNA of that cell, and then we can sequence that DNA and discover the protein target of our lead compound. So this is what it looks like when it's all turned on. The hook, bait, and fish are all jammed together. But there's a problem. It's really difficult to get the hook, the bait, and the fish to all be together. It's not as trivial, it's just, puzzle pieces coming together. So this is kind of where my research begins. So my research goals, I have a big one, or my group did, I guess I should say, is to develop more effective drugs for cryptosporidiosis. We have a medium goal, which is to identify the protein targets of MMB006169, our lead compound, using the yeast 3 hybrid method. And finally, our small goal is to find a site on our lead compound where we can attach the linker, that big molecule, affecting our lead compound's bioactivity against C. parvum. So, to see this pictorially, this is our medium goal right here. We want to find out more about this interaction between the protein target and our lead molecule. Before we can do that, though, we need to do our small goal, accomplish our small goal, which is to bind the linker to our lead compound. And that's not really easy to do. So again, we have to find a site on our lead compound where we can bind that linker without affecting the lead compound's activity against C. parvum. Another way of putting this is we don't want the IC50 value to change. And if you've forgotten the IC50 value, it's just the concentration of our lead compound necessary to inhibit 50% of the pathogenic activity. Okay, so this is our original compound, MMB006169, our lead compound. As you might see, there are a lot of different areas on this compound where we can attach a linker of some sort. Lots of open carbons. We can go here, 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 these. There's a lot of different spaces. But the problem is you can't just throw molecules on top of other molecules because the structure changes of your original compound, and that can potentially change its activity or its IC50 value. So we have to come up with a solution as to how we're going to find a specific site on our lead compound to attach that linker to. Now, some of you may be thinking at this point, well, let's just throw the linker on all the sites and see what happens to the IC50 values. There's a problem with this. The linker, again, is really, really big. It's really difficult to attach it to these sites, and it's a waste of time. Because all you really need to know is how this carbon is going to bind to another 2 or 3 atoms out, if that makes sense. So we're not going to use this method. We're not going to just attach the linker to these where these red arrows are, because it's a waste of resources. So our second solution is to attach small functional groups, just a couple atoms, to each of these locations, and to send them in for biological testing and to see what happens to the IC50 value. This is going to work, let me tell you. Maybe. So the steps to determine where to attach that linker are, again, pretty basic, or not. We're going to synthesize analogs of the lead compound. We're going to validate the structure of those analogs. And then finally, we're going to send them in for biological testing and to determine the IC50 values of each of those analogs. So here we go. We're going to synthesize an analog. So an analog, in case you don't know, is just a compound that is very similar to your original lead compound, but it differs maybe by a functional group. So we can see this here. We have our original compound, and then we have our analog. And you can tell we've added a little methyl group right here, and that's what makes it an analog. So what I did when I was in St. Andrews this past summer, I synthesized 9 analogs total in 4 weeks, which, depending on your point of view, can seem really slow or really fast. For the purposes of this talk, I'm only going to focus on one analog that I made, which we will conveniently call analog A. And again, what makes it an analog is that it has this methyl group on it right here. Okay, so this is our reaction scheme. So, as Charlie talked about before, synthetic chemistry can seem easy at first. You just throw in a bunch of different ingredients per se, and then bish bash bosh, it all comes together, and you get your analog from your starting material. As it turns out, it's not quite that easy. You have to really know how all these individual components are interacting with each other to determine the final product. So this is going to be step 1 for us. We're going from the starting material to the intermediate, and then we're going to have step 2, which involves going from the intermediate to our final compound. So here's step 1. Essentially what happens is you have a benzylamine, this thing right here. The nitrogen acts as a nucleophile. It's going to come in and attack this spot right here, where the chlorine is attached to the carbon. It's going to attach this carbon, which is electron 4. As you know from organic chemistry, or maybe you don't know, as some of us have a hard time remembering, the electron's going to be kicked up to the nitrogen, and then the nitrogen is going to kick the electrons back down until the chlorine comes off. And then you're going to have this final product with an NH2 instead of an NH. So to get rid of the extra NH2, so we start with this here, you're going to use a base of some sort. In our case, we're going to use triethylamine, and that's going to deprotonate one of the H's here, and then you're going to get your final, or your intermediate, I should say, compound. So step 2 is really quite similar. You start with your intermediate again. You have your NH2, which acts as a nucleophile. It's going to attack the electron 4 carbon right here. You do some arrow pushing, the chlorine will come off, and you're going to get this product, but again, you're going to have the extra NH2 right here, and that makes a salt. So then you have to wash this compound using DCM and sodium carbonate, and then this will deprotonate, and then you'll get your final analog, which is always a good thing. So again, we've just synthesized our analog. It worked, let me tell you. Now we have to validate the structure of the analogs. So structure validation is just some sort of verification that you have what you thought you had, or thought that you wanted to make. There are a couple different techniques. There are actually a lot of different techniques. The ones we're going to focus on are IR, infrared spectroscopy, and nuclear magnetic resonance spectroscopy, NMR. And then there are some others too, which I used. So infrared spectroscopy is a technique used to determine the functional groups of your analog. Essentially, it works like this. I'm sure Professor Fleshman is going to just roll her eyes the whole time. It's okay. This is a very simplified version of IR, but hopefully it makes a little bit of sense. So when a chemical compound is hit by IR radiation, the bonds start to vibrate. Okay, and just so you know where we are, infrared radiation is right here. So it's like 10 to the negative 4, 10 to the negative 6 meters. It's kind of the wavelength of light we're talking about. So if the frequency of the IR radiation matches the frequency of the vibrating bonds, the IR radiation will be absorbed, and this corresponds to a specific band in the spectrum. And this is just a nice little picture of the IR. It's quite small, but it works wonders. Okay, I apologize in advance for my spectra. I had to take them off a little PDF because I don't have the fancy computer equipment that they used over in St. Andrews. So here's a PDF version of that spectra. So we're going to focus on two peaks in particular. This one right here, it's kind of hard to see, and then there's a sharp peak right at 1580 inverse centimeters. So enlarge this a little bit. We see this is a really broad peak. This corresponds to the NH group right here. Pretty typical. We will also focus on this peak, around 15 or 1,580 inverse centimeters, which corresponds to the aromatic rings. It's hard to tell, hard to say if you're talking about both rings or if it's picking up just one. You just know that you have some sort of aromatic ring going on. So that was for step 1. So that was for our intermediate. This is for our analog A. So this is our final analog that we are going to send in for biological testing. So again, it looks remarkably similar to our original compound, or our intermediate compound. We see maybe something here, we'll talk about it, and we have a sharp peak here. So this kind of maybe looks like a band that corresponds to NH, probably not. NHs are kind of hard to see on the IR sometimes, so we're just going to call this one a wash and assume that doesn't tell us anything important. Then we can look at another sharp peak right here. Again, it's at 1,580 inverse centimeters, which corresponds to aromatic, the aromatic functional groups. So to kind of summarize that and what that means for us, we determined that there is an NH and an aromatic ring system in the intermediate A.1. So check. We also determined there was an aromatic ring system in our final analog. But we can't determine the difference looking at the spectra between our intermediate and our final analog. So essentially, IR was kind of a bust for us. We're going to need to use another structure validation technique. So we're going to use NMR, which, as Charlie said, is kind of the premier technique that everyone uses. It's definitely very helpful. So essentially, NMR allows you to identify specific atoms in your compound of interest. The principle upon which NMR is founded is that the nuclei of atoms are considered magnetic. They're associated with a charge and, most importantly, a spin of some sort. And then when you apply an external magnetic field, the nuclei themselves are going to align themselves with or against the magnetic field. So this is kind of what NMR is based off of. If you want to learn more, take Chem2, and you'll discover the mysteries of NMR. So this is NMR at St. Andrews. It was really one of the most interesting experiences for me was using their NMR for the first time, because you walk into this massive room and there's at least 6 liquid state NMRs, versus here we have just one, a very small room, and they have a really big room, lots of NMRs, and they have 2 full-time people with PhDs in NMR who just sit there and babysit the NMRs for the whole day and take care of them. And it's a lot of work. So the specific type of NMR I'm going to talk about is 13C NMR. 13C is an isotope of carbon. It makes up, as Charlie said, about 1.1% of all naturally occurring carbon. The nucleus is magnetic, so again, that it means that there's a spin associated with that nucleus, meaning that we can use it for NMR. You might be wondering, why the heck can't we use 12C NMR? Because that's the most abundant type of carbon naturally occurring. The problem is the nucleus is not magnetic. There's no spin associated with it, and that means we can't use NMR. Again, if you want to learn more, take BChem2. So again, this is my spectra pulled from a nice PDF, so it's kind of shoddy looking. That's okay. Some things to notice is that we have a lot of noise in our baseline right here, and that is very typical of a 13C NMR spectra, because again, 13C only occurs about 1.1% of the time, so it takes, the machine has to work really hard to pick up a signal of some sort. This peak right here, this really tall one, is my solvent that I use to dissolve my compounded CDCl3. And as Charlie said, as you move to the left, you will be more de-shielded, so that just means that the electrons associated with each of these individual atoms are moving away from those nuclei, versus when you have a more shielded peak or a more shielded atom, it just means the electrons are sticking closer to your nucleus. So we're going to focus just on two peaks. So we have one around 44.1 ppm for CH2, or 44.1 ppm. This is associated with the CH2 right here. And then we also have one that's really, really shielded at 19.2 ppm, which is associated with our methyl group. So this, again, is for our intermediate. This is after step 1. So this means that a reaction probably worked. So we're seeing the CH3 and the CH2, versus in our starting product, we didn't have that. Okay, so this is our actual analog, our final analog. Again, you can see that the baseline is quite noisy. That's okay. We have our CDCl3 again. This is the solvent I used. A point to note is, like in our other spectrum, there's kind of a mass of peaks right here. Those all kind of correspond to the aromatic carbons in my final compound. The problem with determining which carbon lines up with which peak is that there's so many of them, and you really can't do it by hand. So you have to use a really high-tech, maybe not that high-tech, computer program. I use ChemDraw. Thank God for ChemDraw. And from ChemDraw, they can kind of predict what spectra go, what peaks go with which carbons, and from there you can kind of determine your compound. But again, just to show you two peaks of this analog A spectra, we have our CH2 peak right here, and we have our 19.2 CH3 peak right here. So we're going to summarize our results. We determined that there were the correct number of carbons for the intermediate in A1. I will tell you this is true. I counted them all. There's, I think, 16 or something like that, and we had 16 carbon peaks. We determined that the CH2 and CH3 carbons were indeed there. For our analog A, our final compound, we determined, or actually I determined, I should say, that there were an extra number of carbons in our peak, which just meant that maybe there was some sort of contamination when we took our NMR spectra, when we took our sample. So we need to verify our analogs again using some other sort of validation technique. I will tell you that I use infrared, NMR, and mass spectrometry. The summer before I also used melting point. Whenever you publish a paper of some sort like this where you make a new analog, you need to have, I would say, at least maybe 3 or 4 different sorts of structure validation techniques to be considered legitimate. So we've synthesized our analogs. We've validated the structure of the analogs. A little hand waving there, but we did validate them, I'm sure you. And now our final step is to biologically test the analogs to determine the IC50 value. I'm not going to do that. My lab group does not do that. We send them over to the University of Vermont. There are a couple groups out there that we work with who biologically test them and determine the IC50 values. So for each of the analogs, we find an IC50 value and we compare it to our original compound's IC50 value. And if these two IC50 values are similar, that means that wherever we attach that functional group on our analog, that might be a good place to attach a linker. So once we know where to attach the linker, we can build our bait component just to show you what that is. So once we know where the linker is, we can attach, or once we know where to attach the linker, we can attach it to our compound and synthesize the whole bait component. From there, we can use the yeast 3 hybrid method to find the protein targets of MMB006169, our lead compound for the potential drug. And then finally, we can use this knowledge to build better drugs, which is always a good thing. So some acknowledgements. I would like to thank the Westwood group over at the University of St. Andrews, particularly Professor Nicholas Westwood, who was really, really generous in letting a not lab savvy student come into his lab and mess around and really learn how to, function somewhat well in a lab. Also, Dr. Fanny Schrander, she was kind of the person who really guided me through, making these analogs and testing them and, learning how to validate them quite well. So thank you, Fanny, for being so patient. The faculty and staff, especially the NMR people at the University of St. Andrews, the Ward and Houston groups, those are the groups that are biologically testing our compounds over in Vermont. The Mellon family, who funded me to go to St. Andrews, the Sarah Quant and Thomas A. Acuary family, who also helped me get to St. Andrews, Professor Stephen Debrett, who let me muddle around in his lab as well, and has given me a lot of freedom just to kind of do whatever I wanted to do at Lawrence and in his lab, which is nice. Professor David Hall for teaching Chem 680, chemistry department collectively for being so supportive, and my family and friends, maybe some foes too, but we won't talk about that. And this is my lab group. So this is my professor, this is Fanny, and this is me in case you can't tell. But yeah, there we go. Questions? Yes, Gus.
Gus, organic chemistry 2, so I have a question about the synthesis of the A1 intermediate.
Yeah. This is dumb. Let's do this. What is your question?
So I'm wondering, you had the 2 chlorines, and there was one chlorine that was between the 2 nitrogens, and one that you were actually substituting with was the one that was not between the 2 nitrogens.
Yes.
Why that chlorine and not the other one?
Well, that's a great question. I've asked my lab group that several times, and I've always forgotten. And I've asked Professor Dever several times, and always forget. But I verified this with him yesterday, in case such a question would come up, in case you were prompted to ask this question. Essentially, what we could look for, so if you, okay, yeah, that's probably easier. Will you be able to see it though, is the question.
Yes.
Okay, so this is your compound, or my original compound, and then from there you can draw a nice resonance structure. And essentially what we determine is that this compound right here, if you continue to draw all the resonance structures, you're going to have a lot more, there's more resonance structures than if you took off this chlorine first, which had less resonance structures. So that's why this original compound, or why we took off this first chlorine first, versus the other one.
So to clarify, that second thing you drew is after attack, right? So
Yes.
so draw a bond
Oh,
from that
sorry.
carbon to a nitrogen. No, the chlorine's still there.
Wait, what do we do?
Chlorine's still there, and then there's another bond from that carbon to nitrogen, right? It's a tetrahedral intermediate.
Okay.
That bottom carbon
Yeah.
is a tetrahedral intermediate, right?
Right.
So after we attack with the nitrogen, there's a bond, there's a carbon-nitrogen bond, and the other carbon-nitrogen bond.
Right here.
There you go.
Sorry, I was like,
Right, and so when that attacks, we get that angle. That's easier to spread out that
yeah.
than the other.
So more resonant structures, more stable. That's
So-
what we're going for.
So you didn't see any of the other products?
No,
Okay.
it was always just this one. It was just kind of this blind luck that it worked out well. Some of the time. question too. So, if you've taken organic chemistry, you're kind of taught not to look at all the spectra below that point. Because it's known as the secret region, and it takes a lot of work to kind of delineate what those even mean. So I just didn't look at them. And if you look at my final compound, it's pretty unexciting.
Yeah.
You have, so you can't even really tell unless you're using NMR the difference between that intermediate and the final one. So yeah, you just have some aromatic rings, which are going to show up at the 1580 spot, and you have the NH groups, which may or may not. This is a secondary amine.
Yeah, I was talking about more specifically to look if your chlorine is still there, see if there might be a carbon-chlorine stretch.
I don't, yeah.
What other bond in the key does CHs, right?
Yeah,
Where does CHs show up?
before, it's like 3,000.
About 3,000?
Yeah.
So those P's and then IR that you use when you're in the
Oh yeah, that's it.
Probably CHs.
Check. Yeah, try them.
So since I had like 6 NMRs, did you try to do nitrogen NMR at all?
No. The NMR, I basically stuck with, I did 1H NMR and then 13C NMR, and I did a couple 2D NMR, not a couple, but multiple 2D NMRs, so that was pretty much it. It was really expensive to run, and my lab group had to be really conscientious about how many NMRs we were running. And as a student who didn't know what she was doing, she had to run a lot of NMRs, so didn't want to rock the boat. Yes.
Can you talk a little bit about the 2D NMRs?
Oh, okay, so the 2D NMR, essentially, you combine your 1H NMR spectra with your 13C NMR spectra, and from there, depending on what experiment you're doing, you can see which hydrogens, so there are hydrogens on this ring all around, and these rings right here. You can determine which hydrogen is associated with which carbon.
HNQs?
Yep. It's actually kind of complicated to do if you don't have a good carbon spectra, and some of my spectra were a little shaky because I was using a very small amount of sample to take my spectra with, to take spectra. So yeah. So the really saving grace for me when determining like which carbons were associated with which hydrogens was ChemDraw. And from there you could kind of determine which was which and have a general pattern of, which carbons were with which hydrogens for all my analogs. Yeah.
So extra piece, the carbon 13.
Yeah, I'm not sure about those. I don't know.
Just an impurity?
I think so. Well, I took those spectra a few times, so maybe it's not an impurity, but then again, my NMR tubes were not always the cleanest. So
I guess maybe a better question is, do you have any assurance of purity with any other technique?
yeah, we did mass spectrometry,
Okay.
which was really, as Charlie kind of shows, it just if you have a compound of some sort, you can determine its molecular weight, and you can verify really, really closely, like to the 100, 1,000, how close your analog is to the experimental molecular weight of your compound.
And so then that gave you exact mass to your product, so you definitely get your product?
Yeah, definitely got it.
Did it say that you only got your product?
For most of them, yes.
Okay.
There's some that were a little shaky on, but yeah. So I think I sent 9 analogs over to Vermont. They're testing them. So we'll see what happens. Yeah. Grant's waiting to go, so I'm just gonna sneak on.
All right, let's take it.
Yeah.